अब हम बात करेंगे प्रोसीजर या स्टेप्स विच आर used or which are going to be performed for ELISA test. As we earlier talked about coating of the wells, we have 96 well plate. That plate has to be coated with the antigen or with the antibody. For that purpose, we already have in our materials the antigen, the specific antigen. So 100 microliter of antigen specific antibody diluted in buffer. यहाँ पे हम जो बात करेंगे प्रोसीजर की, वी आर गोइंग टू इमोबिलाइज और वी आर गोइंग टू कोट द वेल्स विद द एंटीबॉडी so that this antibody can detect the antigen in the sample. So for that purpose, 100 microliter of the antibody which is diluted in buffer A is going to be added in each well, and then this plate is covered with a plastic film or aluminium foil, or sometimes the plates are provided with the lid. So cover it with the lid and incubate it at 4 डिग्री सेंटीग्रेड ओवरनाइट. कवर करना इसलिए ज़रूरी है ताकि रेट ऑफ इवेपोरेशन की वजह से जो हमारी वॉल्यूम लॉस ना हो, और चूँकि ये वेल्स बहुत स्मॉल होते हैं, इसमें जितनी वॉल्यूम भी हम यूज़ करेंगे माइक्रोलीटर्स में होगी. एंड इफ़ वी आर नॉट कवरिंग द प्लेट देन वी लाइकली पॉसिबल चांसेस हैं कि हम अपना सैंपल लूज़ कर दें और जो पर्पज़ है वो फुलफिल ना हो सके. So overnight incubation at 4 degrees centigrade is going to coat the plate or is going to coat the wells. Next day we have to wash the antibody or the coating solution. For that purpose we can use the microplate washer, but microplate washer is not available in all the labs, so we can manually wash the plates as well. For that purpose we have the washing solution or washing buffer B. That buffer is used for 3 times washing of the plate, and it can be done by starting with the immersion or inversion of the 96 well plate on a sink, so that our buffer A containing the antibody solution is going to be lost. Then we are going to add the buffer B and we are going to leave it for some time on the bench, on the table. This is the time of incubation, so that any kind of traces are going to be dissolved in the washing solution. Then we are going to discard the washing solution again by inverting 96 well plate over the sink. After inverting or after emptying the wells, we have to tap this plate 2-3 times, gently tap 2-3 times on some of the soft paper tissues, just like this. And the purpose of this inverted tapping is to get rid of any residual liquid. Don't be very hard in tapping to lose anything or to break the plate. This washing step is repeated 3 times so that to ensure no traces of buffer A are left behind. Now the 96 well plate is coated with the antibody. The next step is addition of the sample. Now this sample contains antigen or do not contain antigen will be discovered or will be reported after the ELISA test. We have the 96 well plate. We are going to load the sample, and the sample volume is 100 microliter of the test sample, which is diluted in buffer B, is added. The volume is 100 microliter, and then after covering the plate either with the lid, with the plastic film, with the aluminium foil, we have to incubate it at room temperature for 2 hours. After 2 hours we have to perform the washing, manual washing, as we did earlier at step number 2. Now this is the time that incubation time के बाद एंटीबॉडी एंटीजन कॉम्प्लेक्स बन चुका है. वी हैव टू ऐड आवर कॉन्जुगेटेड एंटीबॉडी पर ऑक्सीडेज कॉन्जुगेटेड एंटीबॉडी. इसको मैं अगर सिंप्लीफाई करूं, वी हैव टू ऐड आवर प्राइमरी और डिटेक्शन एंटीबॉडी. दिस एंटीबॉडी इज diluted in buffer B. 100 microliter volume is added, and then again an incubation is required for 1 hour at room temperature under covered condition. यहाँ पे हम specific antibody use करेंगे to perform direct ELISA. अगर तो direct ELISA use करते हैं, we don't need to perform any secondary antibody incubation period. Otherwise, we have to add an additional incubation step. So once the antibody incubation period is over, this step number 4, again the washing step comes. As discussed earlier, we have to perform 3 times washing so as to remove every material or any traces of the antibody solution. Now the enzyme is there, the conjugated enzyme is there. Now the time is to develop the color so that we can detect the presence or absence of any antibody. 100 microliter of buffer C or chromogenic substrate is added. This substrate is going to be processed by the enzyme which is part of the antibody, and then the color change will appear. For that purpose, we have to incubate the plate for 15 minutes in the dark. 15 minutes can be adjusted unless a suitable color is developed. It can be 10 minutes, it can be 20 minutes, or likewise. Once the required color development has occurred, then we have to stop the color development. For that purpose, we add the reagent D to the wells and we incubate it for some time so that the color development should be stopped. For every well, the time of incubation after the addition of sulfuric acid or reagent D should be same. That variable intensities of the colors can be seen in the plate, and that can be determined using an ELISA plate reader.